Chapter 11, Mendelian Genetics. This PowerPoint will briefly describe the basic vocabulary and a little bit of the history of Mendelian genetics. So let's start off by talking about Mendel. He was an Austrian monk in the 1800s. He studied math and science, and he did not agree with the current idea of the blending concept, which I'll talk about on the next slide. He carried out many experiments, but he found that the peas, the common garden pea, worked the best because he could control which type of pea plant bred with a different pea plant. So he could control breeding amongst the different pea plants. And their generation time was relatively short, so he can get many generations in a short period of time compared to animals. He is frequently known as the father of genetics. The blending concept. The blending concept is the idea that offspring from organisms are a combination of mom and dad. So that would give us offspring that were a blend of mom and dad's characteristics. So if you had a blonde-haired mom and a black-haired dad, this would result in brown-haired children. Of course, then the brown-haired children would breed with other brown-haired children, and eventually all humans would simply have an intermediate or like a brown color shade of hair, which we know that's not true. We have many different colors of hair. So if you were to look at all the characteristics in humans, if we were to breed and select, I don't want to say select, but we would tend to have our characteristics change or blend into this intermediate characteristic, we would all kind of look the same. We would be the same height, we'd have the same IQ, the same eye color, skin color, all these things. We would look very, very similar. So Mendel, he experimented with several types of plants and animals and determined the best subject for this lab was the garden pea. So although he is famous for his peas, he did breed other species. What he found, like I said earlier, was that it had a shorter generation time and breeding could be controlled. And what he did was he picked traits that had two forms. So that term form means like two versions of that gene, like blue eyes and brown eyes. So those are referred to as allele. So he picked the following alleles. He picked green versus yellow, tall versus short, wrinkled versus smooth, purple flowers versus white flowers. There's actually many other characteristics he looked at with peas, but those are some of the main ones that we'll talk about. So what is a pure breed? A pure breed is an organism that breeds true. All right, so what we're basically saying is that organism has the same copy or same alleles for a particular gene. So a purple flower plant crossed with another purple flower plant will always have an offspring that are purple flowers. It's like if you take a poodle and breed it with a poodle, you're going to get poodle puppies. If you get something else, somebody wasn't pure. All right, so since he could control who he was breeding with who, because plants are a little bit easier to do that with, he would cross them over and over and over with the same expressed phenotype, where he then would, I don't want to say guess, but the probability of them not being a purebred was quite low. So he kept breeding them over and over and over again until he got pure lines. That generation he then named his parental generation. Let's talk about complete dominance. So when he cross-pollinated the parental short plants, so we'll say little t, with the parental tall plants, big T, all the offspring were tall. So we would expect our offspring, our F1s, to be big T, little t. So what we didn't see is like a medium or an average height pea plant. You either get a tall pea plant or a short pea plant. Since this was complete dominance, the dominant allele, big T, tall, completely covers up the recessive, the short gene. in this example. So like we had said, the blending concept would have dictated that the offspring would all have been in the middle, okay, middle height. Now the first generation, he called the F1 generation for filial sons, which means in Latin, sons or daughter. So here is a diagram of what we're looking at. We have our parental generation. Whoop, wrong button. Parental generation right here. So we have big T, little t, and of course when we go from being diploid to haploid by meiosis, the possible gametes that we can form from big T is only big T, and little t, we only have little t's to give. So when we cross them together and we get fertilization, our F1s are all big T, little t, and as we can see, they all appear, our phenotypes, they're all tall. Our genotype, that's the genes that they have, they're all big T, little t. So there's the genotype there. So the F1 generation are heterozygous in our example. And another word for that would be hybrids. So they contain one copy of each of the allele for that gene. Now a monohybrid cross, which you already did in class, that has to do with just one characteristic at a time. Now we also have talked about dominant versus recessive. It's not completely random as to which characteristic we decide is dominant and which one is recessive. I just don't pick, oh, I want tall to be dominant, so I'm going to give it a big T. We actually did the cross between our parentals and saw what our heterozygous or hybrids look like, and they all presented themselves as tall plants. So we now know that tall is the dominant gene, and that is why we give them the big T, because dominants are represented by the uppercase letter, and recessive is represented by the lowercase letter. So let's look at the vocab words genotype versus phenotype, and I've already been talking about this throughout the presentation. So our genotype is the actual genes the organism has. So we've been talking about P height, so big T, little t, that is the P's genotype. The phenotype then is actually what we see when we look at them. When we look at those pea plants, big T, they're tall. Big T, they're tall. Little t, they're short. So what we're seeing over here is the phenotype. What we are seeing right here, and we actually don't see that visibly, but we could say the genes they have, they contain inside the nucleus, that's the genotype. So that's the difference between phenotype and genotype. Now in the second generation, Mendel took two of the offspring from the first generation. So we have our hybrids here, and we put them in that Punnett square in possible combinations. So big T, little t, big T, little t, and then we have this big T and little t. So that's the possible combinations that we have. That would be the genotypes. If you look at the ratio of our genotypes, for every one big T, we have two big, let's go back. We've got two big T, little t's, and a little t. Now our phenotype ratio, that's what we see. And when I look at the offspring, 3 plants, or 75%, are tall, and then 25% or 1 plant is short, which gives us our ratio of 3 tall, 1, 2, 3, to 1 short. Now, when we look at his results, we ended up getting that short phenotype back when we bred our F1s to get our F2s. But more importantly, a lot of people are doing these Punnett squares, and they're wondering, if Mendel is the father of genetics, why are we naming these squares after Punnett and actually it's after Reginald Punnett. He was actually one of the scientists that discovered Mendel's experiments. And up to that point, nobody really understood Mendel's work. Mendel was a genius in his time, and, he just wasn't understood by his colleagues. And it wasn't until later, I mean, we're talking quite a few years later, that Reginald Punnett, who was a statistician, so he worked with numbers, actually ran across Mendel's data and was able to understand what he was showing with his experiments and able to put it in a nice little chart that could easily describe that process of undergoing, meiosis and separating out the different alleles and coming back during fertilization to get possible offspring. So that is why they are called Punnett squares and not Mendel squares. Mendel's law of segregation. So each individual has 2 copies of each trait. This is referred to as being diploid, one from mom, one from dad. These 2 copies then segregate or separate during gamete formation, and this is called the law of segregation. So basically what it's referring to is, remember when we bound our homologous chromosome, we paired up, and then as I referred to the divorce, we divorce our homologous chromosomes. One goes to one pole, one goes to the other pole in the cell. Mendel called that his law of segregation. So what he was saying was that each gamete will only have 1 copy of each of the trait or 1 of the homologous chromosomes. In order to get back to being diploid, you will have fertilization. So you'll have the egg and sperm come together, and then the zygote, which is the fertilized egg, now will have 2 copies of that chromosome. They'll be homologous. This is where I ended the original flipped classroom presentation. I wanted you guys to practice your homework problems, which you have and you've turned in, and I will be handing those back for you to check your answers on Canvas. Do make sure you do that so that you do, in fact, know you're doing them correctly. I do want to continue on a little bit further. We did talk about probability in class, but I know one particular class we did not get to fully finish that lesson, so I am going to add it here. So we're talking about probability. We're looking at the possible offspring you are experimenting with. We're looking at what's the probability of getting this type of offspring, which is kind of like what they do with race horses. They're constantly looking at past lines and figuring out the probability of getting that next grand champion. So what we're going to look at is what is the probability of 2 specific gametes that will come together to produce the desired zygote or that grand champion. The statistical relevance to find out whether the outcome is what we expected or if something else is driving that outcome is called a chi-square test. So the laws of probability, what we're going to look at is there's 2 things. We looked at our 1 coin flip and we did our experiment with our 2 coin flips. So the first one will be, what is the likelihood that the event will occur independently? So if you roll a 6 on your first toss, does it affect the chances of not getting a 6 on the second? We've all rolled dice. Just because you rolled a 1 doesn't mean next time your chances of getting a 1 are any different than the first toss. So first of all your possible outcomes have to equal 1. So the chance of rolling a 6 is 1/6. The chance of rolling a 3 is 1 in 6. The chance of rolling a 4 is 1 in 6. And when we look at all those, 1 in 6, 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 and 1 in 6, which is the chance of rolling a 1, 2, 3, 4, 5, or 6, they add up to 1 because we know when we roll that die, you're going to get an answer. You're going to get some number. So we have what's called the and or rule. So if you want to know the probability of some 2 events happening together, you're going to multiply your odds. For example, if you want 2 children, you want the first one a boy and there's a keyword and the second one a girl, the odds of having the boy is 1/2. And then the odds of having a girl are 1/2. So we're going to multiply those. That gives us 1/4. You need to remember how to do your fractions. So with fractions, you multiply the top, which is 1, and you multiply the bottom, which is 4, or 25% chance of getting a boy and a girl. Now, what if I wanted 2 children, the first a girl and the second a boy or a girl? We know the chances of having the first child a girl are 1/2, but we have this and, which is going to go here, so we're going to multiply that with our boy or our girl. So we have to figure that one out. So there are 2 possible outcomes for the second child. Scenario 1 is you have a boy, and we know that the odds of that is 50%. So we have that first one here is a girl and a boy. So we have 1/2 times 1/2, which gives us a 4th. And then we have scenario 2, where we have, again, that girl is the first child, so that's 1/2, and then another girl, which is 1/2, and there's our 1/4. So we have this scenario 1 or scenario 2, so we have to add those. And when we add fractions, remember the bottom, the denominators have to be the same, and then we're going to add the top. So 1/4 and 1/4, and let me just get this out so it doesn't confuse you, is 2/4, and then I reduced it to 1/2. So then we have to look at this as our probability of getting a girl and then a second being a boy or a girl is 1/2. Now the chi-square test tells you whether your actual results are within an acceptable range of the expected results. So we've all flipped coins, and there's been times where we've maybe have flipped almost 10 heads in a row, and it's like, well, is that pure chance or is something going on here? So that's what we're asking. Is something influencing the data? All right. A couple things we need to know in order to do our chi-square is our degrees of freedom, and that's the number of factors in a situation minus 1. So with your coin flips, you could have gotten a heads or tails with 1 coin. So our degrees of freedom, our n, equals 1, because we have 2 possible outcomes minus 1 that gives us our 1. So when we determine our chi-square value, what we're saying is, or what we're questioning, I should say, is our hypothesis correct? Our chi-square value is an acceptable number in which the probability of that occurring simply by chance is 1 in 20. So what we're saying is there's a probability that this would happen 1 out of 20 times we've done that particular experiment that we're looking at. So if the number is, for instance, if we only have our degree of freedom being 1, anything under 3.841 would be acceptable. Once we start getting over 3.841, then we're going to say our hypothesis is null, meaning that it's not correct and something is going on here. So you either need to reevaluate the experiment, rerun it, or try to determine where the observed outcome is being changed or being altered, that is not allowing for your hypothesis to be correct. So this concludes the presentation. I just wanted to put a reminder out there that I am available during period 2 and after school on most days. So if math is an area that you struggle in, please feel free to get help. We will be starting the second probability lab later this week.